That is what happens through beta 2. Because it acts in a smooth muscle, the things that happen here are different. In today's episode, we're going to deal with something that is very important and something that confuses students a lot. So I've simplified it and made it in such a way that there is a lot of basic concepts that help you ease through this topic. So before we go on to this topic, if you haven't watched the previous episode, please go do that. I put the suggestion over here, and that is very important because understanding that is a basic for understanding this topic. So if you haven't watched that, please do watch the previous episode about the G protein coupled receptors and how they act, because that is a fundamental principle that we're going to make use of in this episode. So we'll start off with today's episode. So we got the sympathetic and the parasympathetic nervous system. So people get confused why in some areas there is bronchoconstriction, in some areas there is dilation, right? Some areas it results in again vasoconstriction or vasodilation, while in some other areas there is increased secretion. So people get very confused regarding that. And to remember the receptors, which is actually very simple once you understand the concept. So we got the muscarinic receptors and the nicotinic receptors in case of a parasympathetic nervous system. When we talk about the sympathetic nervous system, we got the alpha as well as the beta receptors. I hope at least that much is clear to you. We got the nicotinic and the muscarinic receptors in parasympathetic nervous system. In sympathetic nervous system, we got the alpha and the beta receptors. So just take a look at what I'm going to do over here. When I'm talking about the muscarinic receptors in parasympathetic nervous system, there is M1 to M5. So I'm going to do a sort of a zigzag over here. I'm going to write M1 over here. Next I'll write M2 over here. And then M3 on top again. M4 beneath it. Where would M5 be now? It will be above this. Okay, so you see what I did? I kind of did a zigzag pattern over here. M1, M2, M3, M4, M5. Now the nicotinic receptors, I am going to draw it over here, somewhere far away. Now think and tell me why I would have done that. The nicotinic receptor is kind of different from these other receptors we are talking about. That is an inotropic receptor. So rapid opening and rapid closing is what happens when we talk about a inotropic receptor. So that is the nicotinic receptor. So the nicotinic receptor is inotropic. It rapidly opens up a channel and rapidly closes a channel. While the metabotropic receptors are comparatively slower. Even if it opens up a channel, it is comparatively slower to a nicotinic channel that is opened up. One thing, and it also takes longer time to close compared to a regular inotropic channel. So metabotropic channels which are opened up, they are going to take longer to close. So that kind of differences are there between a inotropic and a metabotropic receptor channel, right? So that is nicotinic receptor. These are muscarinic receptors. This forms part of your parasympathetic nervous system. Now when we talk about your sympathetic nervous system, we got the alpha and the beta. Alpha is divided into two, alpha 1 and alpha 2. So over here we got the parasympathetic nervous system, and this side I'm going to represent sympathetic nervous system. So the first thing is alpha 1. So alpha is divided into alpha 1 and alpha 2. So in a similar way, I'm going to draw alpha 1 over here. And I'm going to draw alpha 2 below that, corresponding to these. You see that? So we got M1, M2, M3, M4, M5, alpha 1, alpha 2. The next thing that is remaining is your beta receptor. So that I'll draw it separately below over here. We got the beta receptor. And this beta receptor, there is a beta 1, a beta 2 and a beta 3. Three things are the beta 1, beta 2 and a beta 3. Got it? Okay, so we'll just recap it once more. We got the muscarinic receptors, we got the alpha and the beta receptors. These are metabotropic. On the other hand, the nicotinic receptors are inotropic. That is one question. Another question, M1, M2, M3, M4, M5. Right? So let's just avoid M4 and M5 for now. I'll discuss it later. It has to do with your CNS. When we talk about the monoaminergic pathway over there, I'll be helping you understand these receptors over there. So for now, for our discussion of autonomic nervous system, we'll just go with M1, M3, M2, and in the sympathetic nervous system, we have alpha 1, alpha 2, and beta, which has beta 1, beta 2, beta 3. So now we're going to understand the first set of receptors, which are coupled to G alpha Q. Remember G alpha Q we had done in the last episode. So the first thing you need to understand about this is that M1, M3, M5, alpha 1, these are G alpha Q coupled. These are G alpha Q coupled. Alright, so if you had already seen the previous episode, you would understand now that anything coupled to G alpha Q will result in activation of phospholipase C. Phospholipase C will split up the membrane component phosphatidylinositol biphosphate into IP3 and DHE. The IP3 is going to act as a calcium ionophore, which is going to release calcium from the intracellular system. So there is an increase in calcium intracellularly. Also, the diacylglycerol will activate protein kinase C, and ultimately that is going to act on downstream targets. So what you need to understand is through different pathways, RAF, MAP kinase pathway, it is finally going to result in contraction of the muscle. So the IP3 will result in calcium influx, and DAG also will result in contraction ultimately. So in order to understand how this is actually acting, let's just take a look at the muscle, smooth muscle, and try to understand how it is contracting and how it is different from skeletal muscles. Just take a look at this video. Something you need to understand right now is to differentiate between how a cardiac muscle and a smooth muscle contracts. Whether it's cardiac or skeletal, how that contracts and how different it is from how a smooth muscle contracts. Because as you know, the autonomic nervous system kind of acts on your smooth muscle. Right? So when we talk about the cardiac or the skeletal muscle, there is troponin which is present. And that troponin has different domains. One domain is to bind to calcium. One domain, which is the C domain. We have a T domain, which is binding to your tropomyosin. And one I domain, which kind of inhibits the interaction between actin and your myosin. Right? So C domain is where calcium binds. I domain is where inhibition between the interaction of actin and myosin takes place. And T domain is where it is going to bind to your tropomyosin. So we got the tropomyosin over here. It kind of binds over there. And this tropomyosin traverses an area on your actin filament. So if this is the active filament, right, and if this is the one site in the actin filament which is kind of masked by this. So the tropomyosin troponin complex kind of masks an active site over here. When there is influx of calcium, that calcium is going to activate this so that this tropomyosin complex moves outside. When they move outside, this gets unmasked. So when that gets unmasked, your myosin, which is over here, is now free to attach itself to the active site, and then it will result in contraction. So essentially that is what is happening in the case of a skeletal muscle or a cardiac muscle. But when we come to your smooth muscle, you need to understand that smooth muscle does not have this troponin. It does not have the Z filaments. Instead, it has dense bodies. The arrangement of actin and myosin is not as sequential and striated like them. The kind of difference. So without the troponin, how else is a smooth muscle acting? So that is what we need to understand right now. In a smooth muscle, we have another mechanism which is present, which is through a calcium calmodulin pathway. There is a structure called the calmodulin. So there's a structure like this called calmodulin, and in the calmodulin there are sites where calcium can attach itself. Over here, over here, over here, over here. So there are 4 sites where calcium can attach. So 4 calcium with the calmodulin forms what is called the calcium calmodulin complex. Now this calcium calmodulin complex can go downstream and activate certain things. One of that thing is what we call myosin light chain kinase. It's an enzyme. So this is your myosin light chain kinase. It's a kinase of myosin light chain. And when this calcium calmodulin complex gets activated, it is now going to form a loop over here. You understand that? That is going to activate your myosin light chain kinase. So the myosin light chain kinase and this kind of forms a loop like this, right? When calcium binds to your calcium and forms the calcium calmodulin complex, that kind of activates your myosin light chain kinase so that it can now move and activate your myosin light chain. So the myosin has a heavy chain on top and it has a light chain. So this light chain is activated, right? So this is the light chain. So the light chain of myosin gets activated by this kinase which is present. So myosin light chain kinase activates your myosin, and now it activates. So because of the influx of calcium here, there is activation through the calcium calmodulin complex, activation of myosin light chain kinase, and that activates your myosin. You see how it is different between the skeletal cardiac muscle and smooth muscle. So now that we understood how different a smooth muscle is from a skeletal muscle or a cardiac muscle and how differently it acts, you can get the picture of how anything that is coupled to G alpha Q is going to result in increase in the intracellular calcium, and that is ultimately going to result in contraction of the smooth muscle. Also, when you talk about exocrine glands, for example, over there as well, when the G alpha Q coupled receptors are activated, they will result in some sort of secretion. So the smooth muscle contraction as well as secretory activity of exocrine glands, that is what G alpha Q coupled receptors are doing. So let's take a look at the sympathetic nervous system. Just imagine where contraction is needed. Take a look at the lungs, the bronchus. Is there constriction or dilation? There is dilation. So you know that that cannot be your alpha 1. Right? So it must be something else. So we're done with that. Think about peripheral vasoconstriction. Vasoconstriction. So what should it be? It should be alpha 1, vasoconstriction. What about the eyes, the pupil? Pupil needs to constrict. Should it? No. It needs to dilate. Right? So that dilation is because of the constriction of the dilator pupil. That is something you need to understand. The dilation of the pupil is not because of relaxation. The dilation is because of the contraction of the dilator pupil. So that contraction of the dilator pupil through the sympathetic nervous system is through your alpha 1. So in a similar way, wherever in the sympathetic nervous system there is need for a constriction of the smooth muscle, that is where alpha 1 is coming into play. Another area where alpha 1 is coming to play is finally in ejaculation. So you know that initially the process of erection sexual arousal is because of the parasympathetic nervous system, but the final accumulating act is sympathetic in nature. And that is because of alpha 1. And alpha 1 will result in constriction and it will result in ejaculation. So you must be knowing already how during that phase there will be increased sweating, increase in the heart rate and things like that related to your sympathetic activity. So that is your alpha 1 in the sympathetic nervous system because it is coupled to G alpha Q. Now let's come to the parasympathetic nervous system. Where do you want constriction and motility and things like that? So all the exocrine glands, you know that normally the digestive processes, exocrine gland secretion, everything takes place because of parasympathetic nervous system. So in parasympathetic nervous system we have M1 as well as M3. So for the exocrine glands, mostly they are overlapping M1 and M3 for glandular secretion, especially salivation. Both M1 and M3 are there. And M1 is also there in CNS. So it seems to have some sort of overlap with M1, M4 and M1, M3. But predominantly most other things are because of M3. The smooth muscle contractions and all are generally more because of this. M1 is also present in parts of your esophagus, smooth muscle, but otherwise it is predominantly M3. So you need to understand both M1 and M3 are there for secretions and some sort of overlap are there, but generally most other things are M3. You think of peristalsis, gut motility, that is M3. Secretions, the GI secretions as well as salivary secretion, all that M3, M1. Sweat glands, M3. You know the sweat glands are because of the action of M3. Acetylcholine is released from the sympathetic cholinergic fibers. So sympathetic cholinergic fibers are going to release acetylcholine, and regardless of whether it is sympathetic or parasympathetic nervous system, it is still going to act on your M3 receptor and result in a secretion over there. So that is again M3. I hope this is clear. So if you take a look at this, you can understand how simple it is. So anything coupled to G alpha Q is making things contract. So what you need to do to understand this topic is go to the first video where I'm talking about what are the different effects. So logically thinking, you can understand all that. Now using these concepts, you can say which receptor is working there without even by hearting things. Yes? Okay. So that is about the M1, M3, alpha 1. Alright? Yes. So the next thing, I'm going to skip this and go over here. Beta. Beta is coupled to G alpha S. The whole of beta. That is beta 1, 2, 3, it's all coupled to G alpha S. G alpha S, as we know, is going to result in protein kinase A activation finally. Adenine cyclic AMP protein kinase, and it is going to activate a lot of things through phosphorylation. So when we take the heart, it is beta 1. Beta 1 is also there in the juxtaglomerular apparatus, where it is going to result in release of renin. Alright, so that is G alpha S. So all the things coupled to G alpha S are usually stimulatory in nature. So just take a look at the next video in order to understand what it actually does in your heart. So in order to understand how the alpha stimulatory in the beta 1 is going to affect the heart, you need to understand a thing or two about how the heart contracts, how the cardiac muscle contracts and relaxes. So when you're taking the heart or any muscle for that factor, they have a T-tubule. With the muscle, this is the membrane, the sarcolemma, there is something going into it that is called the T-tubule. In a cardiac muscle, this T-tubule will be going inside, and it's said to be actually axial compared to your other one, skeletal muscle. So this T-tubule is present, and the depolarization which passes through, right? Remember the action potential we had talked about, like this. This action potential is going to keep spreading inside and it will go in. And at that point, it will activate a set of channels over here. And you also have to understand that inside the sarcolemma, inside the membrane of the cardiac muscle, there is something called the sarcoplasmic reticulum. So sarcoplasmic reticulum is endoplasmic reticulum which is there in the muscle. So this sarcoplasmic reticulum is where things like calcium, glycogen are all stored. So it is an intracellular store. So the depolarization curve reaches over here, and that is going to activate the first set of channels. So the first set of channels include a calcium channel. So most often we see that there is a channel called CAV 1.2, caveolin 1.2 channel, and a dihydropyridine receptor channel. So dihydropyridine receptor as well as a caveolin 1.2. So it's called dihydropyridine receptor channel because something called dihydropyridine seem to inhibit this. So that is how it got the name called dihydropyridine receptor channel and caveolin 1.2 channel. So ultimately, because of all these, there is an influx of calcium. So the extracellular fluid, calcium, is going to come inside. Alright, so this channel is supposed to be like this. It has 4 subunits like this, 1, 2, 3, 4. And that is going to let calcium inside. So calcium has come inside. And because of the influx of calcium, that is going to activate another set of channels over here. And that channel is called the ryanodine channel, ryanodine receptor channel. This is ryanodine receptor channel. And just keep in mind that something called a calstabin normally inhibits this. So what did we see? There is an influx of calcium, and that influx of calcium is going to result in activation of another intracellular channel which is present on your sarcoplasmic reticulum, and that will again result in calcium, the intracellular calcium store, to be released. So if we take a look at this under electron microscopy, a fluoroscopic electron microscopy, we can visualize this as a calcium induced calcium spark. So you can see this as sparks and a calcium induced calcium spark. So for a cardiac muscle, extracellular fluid calcium is important. Because of an influx of extracellular calcium, there is a calcium stores released from the intracellular store. So both intracellular as well as extracellular calcium is important, and extracellular calcium is important to elicit this response. So that is one thing that is different from a skeletal muscle. In a skeletal muscle, it is predominantly your intracellular calcium. What else is different between skeletal muscle and this cardiac muscle that I just mentioned? Over there, it is a mechanical coupling. Over there, there is a dihydropyridine receptor, and there is also a ryanodine receptor. So the dihydropyridine receptor and ryanodine receptors are there, but they are mechanically coupled. So every time the action potential reaches over here, activates the dihydropyridine channels, that is going to result in a mechanical activation of Raynaudin channel to release intracellular calcium. So you see how there is no role of the extracellular calcium in the skeletal muscle, but over here there is importance of the extracellular fluid calcium. All these are MCQ questions. The receptor, this receptor over here, this receptor I had mentioned, right? The importance of extracellular fluid calcium, and all these are important. And the location of this sarcoplasmic reticulum. Alright? So that is the first thing. Okay? Okay. And because of this calcium, what is going to happen? This calcium is going to bind to your troponin. Right? Troponin has different binding sites. It is going to bind to the troponin C part. Right? Once it binds to the troponin C part, the tropomyosin which is there, which is masking a site on your actin. Right? So in the actin, the tropomyosin is masking a particular site. Once this is released, right? Once calcium combines over here, this is going to get released, unmasking that site which was attached over here. So once that site has been unmasked, now your myosin is going to bind over here, and that is going to result in contraction. So you get the picture. In a cardiac muscle, there is an extracellular fluid calcium. When there is activation of the dihydropyridine receptor or caveolin 1.2 channels, they are going to result in a calcium influx, and that calcium influx is going to activate the Raynaudin receptor channel. So calcium induced calcium spark is there, and because of that, calcium, the troponin and the tropomyosin complex moves away, unmasking an active site of the actin, to which the myosin is now going to bind, and that will result in your contraction. Alright? Okay. So the amount of calcium is going to determine how much of contraction there is. Okay? Now apart from this, later when you need to relax, the calcium needs to go back into the system. Right? The calcium which was binding over here should go back. And that is a very important step. If that is not taking place, relaxation cannot take place properly. So for that, we have certain channels over here. Have you heard of the term SERCA? It's called sarcoplasmic endoplasmic reticulum calcium channels. Right? So they are calcium ATPase. ATPase. So it is going to be active. Right? Against the concentration gradient, it is going to use ATP and actively transport calcium inside. And the channel is SERCA channel. Sarcoplasmic endoplasmic reticulum calcium channels. Calcium ATPase. Right? And just like I had mentioned, calcitonin over here, there is another molecule which is associated to it called the phospholambin. And this phospholambin inhibits your SERCA. Alright, so Raynaudin receptors are normally inhibited by calcitonin. Phospholambin normally inhibits SERCA. And because of the effect of beta 1, right, which is coupled to an alpha S, the phosphorylation that happens will result in greater activation of this. So greater amount of calcium coming in, and it is kind of going to inhibit this. So more calcium coming in. So what happens? Increased calcium, increased contraction. So your inotropic effect is coming here. So inotropy is one thing that is going to happen because of the effect of beta 1. The next thing that is going to happen is something called the lucitropy effect. That is this active reabsorption of calcium. So it is now going to inhibit this and activate this so that calcium can come back. So this is very important. One of the reasons why when there is excess extracellular fluid calcium, the heart kind of stops in systole. Haven't you ever wondered while learning that here, because of beta 1, there is influx of greater calcium, but such an effect is not seen with that. But when there is only an influx of calcium, extra calcium that is coming in, the heart kind of stops in systole. Why? Because this is not activated along with that. So excess amount of calcium will make it keep contracting until it stops in systole. But if you give a beta 1 stimulation, not only is it going to activate this, it is also going to activate its relaxation. So that is why this is important. Lucitropy effect is also there for your beta 1. Okay. Now apart from this, if you take a look at the heart, we have the AV nodal conduction. And when there is stimulation of beta 1, what happens is that there is a increased transmission, greater speed of conduction. Right? That is what is called the dromotropy. So we have a dromotropic effect. Increased speed of conduction in the conducting fibers, especially the AV node. It delays the AV node. It reduces the AV nodal delay. And I talked about the action potentials. So this action potential as well as the pacemaker action potential will be modified because of this alpha stimulatory. That is going to make influx of calcium and sodium much faster, resulting in faster depolarization. So the heart rate increases because of faster depolarization. So slope of the depolarization changes. Alright, so instead of a pacemaker potential like this, it will now go like this. You see, the same time there is more number of pacemaker potentials. So that is your chronotropy. Alright, so these questions can be asked. Based on this, different options can be given. So a stimulation of beta 1, which is alpha S coupled, will result in inotropy, will result in lucitropy, will result in dromotropy, will result in chronotropy. Alright, and basically the excitability of the tissue is increased, that is bathmotropy. So now we have a pretty good picture of what happens because of beta 1 in the heart. So we already seen what it does in the heart. Apart from that, we have the beta 3, which is there in the adipocytes. So when sympathetic activity is present and you need thermogenesis, non-shivering thermogenesis, that is when beta 3 is going to act on the brown adipocyte tissue. And there is increased lipolysis in the white adipose tissue. So increased lipolysis through beta 3 white adipose tissue, increased non-shivering thermogenesis in the brown adipose tissue through beta 3. So that is the effect of beta 3. Again, that is some sort of stimulatory activity. It's not really any contraction or things like that. And that stimulatory activity is through beta 3. So that is clear. Beta 1 is a stimulatory activity. Beta 3 is again a stimulatory activity. You can understand why that would be needed. You need energy. In sympathetic activation, you need energy. And that energy is provided by lipolysis. Initially, it is carbohydrate. Then, once that is depleted, you need lipolysis also. That is the next layer. And especially when things are cold, again, you need non-shivering thermogenesis. That is sympathetic activation. Am I clear? Yes. So, that is beta 1 and beta 3. We'll just leave beta 2 here for now and come back to it a little later. Next thing is these receptors. So these receptors M2, M4, alpha 2, these are coupled to G alpha I. So that is inhibitory. So whatever this does, this opposes it by inhibiting it. So it simply inhibits the adenine cyclase and cyclic AMP is decreased. This stimulates adenine cyclase. So they are opposing action. So this M2 that we're talking about is present in the heart and that opposes your beta 1's activity. Right? Okay. Now another thing often overlooked by many people is something a little different in M2 receptor. I told about a alpha as well as a beta gamma. Right? Generally all these I mentioned is because of alpha's activity. Right? In M2 especially, beta gamma also has a role. The beta gamma is going to go and activate potassium channel. When potassium channel is activated, what is going to happen? Potassium is going to go out, right? So that will result in potassium efflux, and potassium efflux will result in hyperpolarization, right? So something gets hyperpolarized, rate reduces, heart rate reduces because of the pacemaker potential and the ventricle muscle. You can understand how hyperpolarization, and that will slow things down. Alright? Yes. Along with that, it is also acting through alpha. What about this set of receptors in the sympathetic nervous system? That is where your alpha 2 is coming into play, which is coupled to G alpha inhibitory. So how does that act? That acts both presynaptically as well as postsynaptically. So till now, we've seen all these other receptors which are there in the postsynaptic membrane. But this receptor is present presynaptically as well as postsynaptically. So just take a look over here. This is the presynaptic terminal, and over here we have the postsynaptic terminal. And from here the neurotransmitters like norepinephrine are released, where they're going to act on some postsynaptic receptor, like say for example the alpha 1. The alpha 2 receptor is present presynaptically like this, so that this norepinephrine can act on the alpha 2 and thus through alpha inhibitory inhibit its own action. Is that clear? Now instead of alpha 1, say it was beta. It's still the same thing. Instead of activating this same neurotransmitter is going to go inhibit its own activity by coupling to G alpha inhibitory through the alpha 2. So that is one way by which it is acting presynaptically and inhibiting itself. Another way is for it to act postsynaptically as well. So when you take a look at the energy metabolism in the body, when we talk about the sympathetic system, you need greater energy. You need more glucose available. Gluconeogenesis should take place. The glycogen should be cut down to release glucose. Glycogenolysis should take place. And all these things are mediated through your beta. So beta stimulatory. I have mentioned how it is stimulatory. So it will activate all these processes by phosphorylation in the liver especially. And in the case of your adipose tissue through beta 3, it's going to result in lipolysis. That is the second storage. After carbohydrate, that is the next line. So that way, all the energy metabolism requirements are met. But at the same time, you should make sure that apart from the catecholamine growth hormone and the glucocorticoids that act, the other thing which is like acting opposite to that, the insulin should be reduced. So you know how insulin is opposing this activity. It tries to take glucose, excess glucose, and store it in tissues. So that is what the insulin does. So you don't want the glucose to be stored right now. You need energy right now. So insulin needs to be inhibited. So which among this in sympathetic system is inhibitory? Alpha 2. So that is how alpha 2 mediated insulin decrease and beta mediated stimulatory, glycogenolysis, gluconeogenesis, lipolysis. Understood? So that is alpha stimulatory and this is alpha inhibitory. So it's that simple. Wherever there is inhibition, there is alpha inhibitory. Wherever there is stimulation, alpha stimulatory. Wherever there is contraction needed, there is alpha Q. Now apart from all this, we got your beta 2. We had left it out earlier. We had talked about beta 1, we had talked about beta 3, we had left out beta 2. Why did I leave it out? We'll come to understand right now. When we talk about sympathetic nervous system, we told that vasoconstriction, peripheral vasoconstriction, alpha 1. Yes, the constriction of the dilator pupillae, alpha 1. Ejaculation, alpha 1. But what about areas like the bronchus when you need bronchodilation? What about the GIT where you need to inhibit the peristaltic movement? The motility should be inhibited. So where does that come into picture? So I told that alpha 2 inhibits this alpha stimulatory action. But we're talking about smooth muscles over here. We're not talking about cardiac muscle, we're not talking about skeletal muscle for it to be acting through this S, to inhibit through I. So this is no longer coming to the picture now. Now we're talking about the smooth muscles, and smooth muscles was G alpha Q coupled. Now that is where you need to understand that these beta 2 receptors are specifically located in smooth muscles. Now then comes the next question. When beta 2 is activated, it is still activating through alpha stimulatory. Then why is it relaxing a smooth muscle? Now for that, you need to go back to that earlier diagram I had drawn about the smooth muscle, contraction in a smooth muscle. So we'll just go ahead over there. Now we need to understand how beta 2 is kind of inhibiting everything. Beta 1 is alpha stimulatory, beta 3 is also alpha stimulatory, beta 2 is also alpha stimulate. But why is it that beta 2 kind of results in relaxation while beta 1 and beta 3 activates things? Because it is ultimately the alpha stimulatory, which is going to result in activation of adenine cyclase, cyclic AMP, protein kinase A, which is going to phosphorylate something and activate things. You already seen how it happens in the heart. But why is it that beta 2 relaxes things? That is where you need to understand what happens here. We already mentioned how a smooth muscle contracts. It's very different from how a cardiac muscle contracts. Now once we got that picture clear, we need to see how beta 2 is now going to act. So beta 2 will act on the alpha stimulatory adenyl cyclase, cyclic AMP, and that is going to activate protein kinase A, which is now going to phosphorylate something. Over here in the cardiac muscle, the phosphorylation kind of activated your calcium channels. It activated your calcium channels so that more calcium comes in. It activated your circa so that lucitropic effect is there. It activated your conduction, and it activated channels in the pacemaker potential as well as action potential so that the rate of potential is changed. So chronotropy effect is also there. But over here, this phosphorylation is for your myosin light chain kinase. Now once myosin light chain kinase gets phosphorylated, its structure is kind of modified. So even if there is influx of calcium, that calcium cannot activate your myosin light chain kinase through the calcium calmodulin pathway. So this normal process that happens gets inhibited. That is what happens through beta 2. Because it acts in a smooth muscle, the things that happen here are different. It is going to go phosphorylate your myosin light chain kinase, and phosphorylation of myosin light chain kinase will make it difficult for calcium calmodulin pathway to now activate your myosin. So contraction does not take place, and that results in relaxation. So that is what happens with beta 2. And that is how that is different from everything else. The beta 1 and beta 3. It's not that this is different. This is still the same. This mechanism is still the same. But the affected tissue is what is different. And the mechanism inside that affected tissue is what is different. Because smooth muscle contracts very differently. I hope this is clear, and I hope you don't forget this. Because it's a very simple concept, and once you understand this, you can understand how beta 2 is very different. So let's just do a quick recap of all these things and try to understand the difference between a sympathetic and parasympathetic nervous system with regard to its physiological mechanism as well as its molecular mechanism. So when we take a look at the sympathetic nervous system, first thing is we know that the visual field should be enlarged. For that, dilation of the pupil should be there. For that, constriction of the dilated pupil should be there. It is a constriction of a smooth muscle. So which of these is going to be important there? Alpha 1. We know that it is coupled to G alpha Q. So alpha 1 mediated constriction of the dilated pupillae. That is done. When we come to the lungs, the bronchus, we need the bronchus to dilate because we need more air while we're running for your life. Isn't that right? So that is a dilation. So we know that dilation is not alpha 1 mediated. We know that beta 2 is predominantly present in places where you want a sympathetic mediated dilation. Because in smooth muscle, the mechanism is different. So the beta 2 mediated alpha S coupled phosphorylation of myosin light chain kinase is going to result in relaxation. So that is beta 2 mediated relaxation of the bronchus. When you come to the GIT, do we need the GIT muscles to constrict more, peristalsis more? No. So that again is beta mediated. But the sphincters should constrict. And that is constriction alpha 1 mediated. And when we come to the bladder sphincter should constrict and bladder muscle should dilate or relax so that we do not pass urine. That constriction of sphincter is alpha 1 mediated. Dilation of the bladder is beta mediated. Now this is one thing I had not mentioned earlier. When you talk about the urinary bladder, there are conflicting reports. Some say that it is beta 2 mediated, some mention beta 3 mediated, where I have seen many articles where they mention the presence of both beta 2 and beta 3. What you need to understand is, regardless of whether it is beta 2 or beta 3, ultimately all beta is going to act through the same mechanism, and through the same mechanism it is going to result in relaxation. And because I've seen it's a very conflicting report, nobody is going to ask you whether it is beta 2 or beta 3. If somebody is asking you that question, he probably doesn't know that there is something else which is present. So you can outright go to the court and give references based on this. And so that is your bladder. We done with the GIT, we done with the secretions, and we done with vasoconstriction, which is alpha 1 mediated. So that is your sympathetic nervous system. And in energy metabolism, we've seen how a beta related stimulation would result in glycogenolysis and gluconeogenesis, and a beta 3 mediated lipolysis takes place. On the other hand, insulin secretion is decreased, that is alpha 2, and if it wants to inhibit its own activity, that is alpha 2 mediated presynaptic inhibition of its own activity. And in the heart, you need, it's a cardiac muscle, not a smooth muscle anymore. So that is beta 1 mediated alpha stimulation. That is sympathetic nervous system. When you come to the parasympathetic nervous system, do you want the pupil to constrict to dilate? It is mostly near vision, so it will be constricted. That is your constriction. And what is important for constriction in parasympathetic nervous system? M1, M3. So we told most of it is M3. What about salivary secretions? Again, that is G alpha Q coupled. That is both M1 and M3. Exocrine glands mostly are both M1 and M3. GIT, you need peristalsis, you need motility, you need secretions, that is again M1, M3. And when we take a look at the urinary bladder, you want the bladder to constrict now. And that constriction of the bladder should be again this. So that M3 mediated constriction of the bladder. And when we talk about the sympathetic cholinergic fibers which result in sweat gland activation, sweat gland is also a gland secretion, that is also G alpha Q coupled, which is G alpha Q coupled, M3 is most predominant. So that is another thing. And then when we talk about your heart activation is alpha S. So you need something that is alpha inhibitory to oppose that action. That alpha inhibitory is your M2. Now M5, M4 are present in the CNS. Also, some parts of M1 are also present in the CNS. So, are you with me till now? I hope you understood everything. I hope you were able to recap. If you have not been able to, please go back, try to read it again, try to listen to it again if possible, and formulate all these things in your mind. Try to draw it, and once you get a hang of it, you'll never forget it in your life again. You don't have to keep by hearting this ever. Now, one thing I need to specifically say right now is about your M3. So I didn't say it earlier because I didn't want you to get confused. Now, when we talk about the parasympathetic nervous system, earlier reports suggested that the innervation of parasympathetic activity on your blood vessel is very minimal. There are few receptors, but the effect of parasympathetic activity is very minimal. But there are examples where your muscarinic receptors are known to dilate your blood vessel through M3. Now that goes against everything we've done so far. So that's why I didn't want to confuse you earlier. M3 till now was G alpha Q coupled and results in contraction of everything. But in a few places, for example, for erection, there is dilation and pooling of blood there. And that is M3 mediated. That is why you have to understand that it is not only M3 mediated. There's something called co-transmission. When a vesicle containing acetylcholine is released from the nerve terminal of parasympathetic postganglion, it not only releases acetylcholine, there are other substances that are released along with it. One most important thing is your nitric oxide. So both nitric oxide and acetylcholine is released. Nitric oxide is going to dilate it. Acetylcholine is going to act on M3, and now that M3 is going to activate your epithelial nitric oxide synthase and result in increased nitric oxide again, which that is basically how it is dilating. So till now we know that alpha 1 is constrictive for the peripheral vessels, blood vessels. We know that beta 2 relaxes blood vessels in the skeletal muscle. And now we know that M3 in blood vessels, it results in relaxation through the M3, and basically the final thing is your nitric oxide. So I seen two questions based on this. I'll try to post a video separately some other day about co-transmission, because this co-transmission is also present in your sympathetic nervous system. So we'll do that again. So I just wanted that to be clear in case a question like that comes. Yes? Okay. So hope this helps you out, because this was one topic which is very confusing for me, and I was an undergraduate student. And I know I struggled the entire 5-6 years of my life and never really understood this. So I hope this helps you out, and please do revise this again. Yes? Okay. Thank you.